Once again, thank you all for joining the call. Happy New Year to those I haven't said it to already. And welcome to the first Jetson call of 2024. We have two excellent talks waiting for you today. And so without further ado, I will pass over to the first speaker, Manon Laget, who's going to talk to us about giant mesopelagic protists. Over to you, Manon. Manon, you're on mute, I'm afraid.
Okay, sorry. So is the screen sharing okay for everyone? Great. So thank you, Adrian, for the organization. And so to introduce myself, I just finished my PhD last month, and I started a postdoc position like 10 days ago in Halifax, Canada. So I will present some results that I got during my PhD. So, which is about giant mesopelagic protists in the carbon cycle. So I will be quick on this part, but just to remind you, the biological carbon pump is the process of production of particles of biological origin using the atmospheric CO2 via photosynthesis, and also their transport downward from the surface to the deep ocean. So these particles can be transported via multiple pathways, and in particular via gravitational sinking, which is a key pathway of the biological carbon pump. So when particles are exported out of the surface layer to the mesopelagic layer, they are consumed and transformed. So for example, by zooplankton and bacteria. So in particular, mesopelagic zooplankton is known to consume and transform particles. So zooplankton can, for example, fragment large particles into smaller ones, or it can repackage slow sinking particles into fast sinking fecal pellets. And mesopelagic zooplankton communities are extremely diverse because it includes organisms such as crustaceans like copepods, salps, for example, or also protists, such as Rhizaria. Sorry. And a key question of biological oceanography is how zooplankton can impact the gravitational flux. So here I will talk of specific zooplankton group, so Rhizaria. So Rhizaria are a major eukaryotic supergroup, which includes unicellular eukaryotes or protists from like diverse ecosystems, so aquatic or terrestrial or marine ecosystems. In the marine ecosystems, it includes in particular foraminifera or radiolaria. So here I will focus on two groups of strictly planktonic Rhizaria, so Radiolaria Polycystemia and Phaeodaria. So as I said, these groups are strictly planktonic, and they are found from the surface ocean to the deep ocean. So also most of Rhizaria, of planktonic Rhizaria can produce a skeleton made of biogenic silica, and this skeleton can have various structure and ornaments, as you can see on this picture. So on the top picture, you have a Phaeodaria skeleton, and the bottom picture shows the skeleton of two Radiolaria specimens. You also have Rhizaria with no skeleton at all. So Rhizaria are highly diversified protists. Here you have the size spectrum of plankton, which covers several orders of magnitude. And here you have the size spectrum of Rhizaria. So planktonic Rhizaria can cover a large part of the planktonic size spectrum. So they can be as little as 20 micrometers for the smallest Radiolaria, but also these unicellular organisms can reach more than several millimeters in size. So as you can see on all these pictures, their morphology is extremely diverse, and in particular, the shape of the skeleton or of the organic structure. So in this talk, I will mainly focus on large rhizaria, so on rhizaria above 200 micrometers. But there's something that makes difficult to study rhizaria ecology is that these organisms are extremely fragile, and they often break when we collect them with plankton nets. And also, it is very difficult to culture them, so to maintain them alive in vitro, so to have information regarding their life cycle or their physiological rate, for example. By contrast, you can sample rhizaria using in situ imaging instruments. So here you have the size spectrum of various in situ imaging instruments. And in particular, to sample rhizaria, we can use the UVP, which can get images of particles above 600 micrometers. So we can get images of large rhizaria with this instrument. So using the UVP, we could we could know with more accuracy the vertical distribution and the drivers of planktonic rhizaria. So as you can see on this figure, for example, and what it showed is that rhizaria have a wide range of vertical habitat. So from the epipelagic layer down to the mesopelagic layer. And they have a diversity of trophic functions. So on the one hand, in the surface layer, you mainly found radiolaria, which display a mixotrophic behavior. So this means that besides the fact that they feed upon various prey, they can also harbor algal symbionts and do photosynthesis. So you can see the symbionts on these pictures. So all the yellow dots are the symbionts, the algal symbionts of this radiolaria. And on the other hand, the mesopelagic populations, so which mostly include Phaeodaria, are strictly heterotrophic and they are flux feeders. So they feed on the sinking particle flux. And they can store their food in their organic structure, as you can see here. So here, this map shows the first global estimation, which was done in 2016, of large rhizaria abundance from a global UVP5 dataset. So at global scale, this dataset showed that large rhizaria can account for about 30% of mesozooplankton abundance. And in terms of biomass, there also have been a few estimates using the UVP5. So this study showed that they could represent 31% of mesozooplankton biomass in the epipelagic layer. And a more recent study showed that they could represent about 20% of mesozooplankton biomass also in the epipelagic layer. So this study is from 2022. That's a mistake. And this figure on the right shows the abundance of zooplankton according to latitude. So the current knowledge is that rhizaria abundance, so that you have here in blue, increases towards intertropical regions. So this pattern of rhizaria is in contrast with the pattern of other zooplankton organisms that you have here in orange, such as, for example, crustaceans, which are known to be highly abundant at high latitudes. So in addition to their contribution to zooplankton abundance, rhizaria can also have a role in vertical fluxes. So they can contribute substantially to fluxes of organic carbon because they can be associated to large pulse events of particles. And also, large Phaeodaria can form fast-sinking particles by aggregating detrital material around them, and they can efficiently export them to the deep ocean. And their skeleton is supposed to be to have a ballasting effect. However, heterotrophic Phaeodaria, so as I told you before, they are flux feeders. So they also consume sinking particles. So they can also contribute to flux attenuation below the surface layer. So for example, there was a local study which showed that they can intercept about 10% of particle flux in the California current. But there is no assessment of this role on the gravitational flux at global scale yet. So to better understand the distribution of Rosaria biomass and their role in fluxes, we first need to convert their abundance to carbon biomass. And for that, we need reliable conversion factors, which were not really present in the previous biomass estimates that I showed you. And a way to convert the size to carbon biomass is to use allometric relationships so that we can use this relationship for a global extrapolation. So in the past, an allometric relationship, which converting the cell size to carbon biomass, was established for a variety of protists, but this relationship does not include any Rosaria. So we can wonder whether this relationship would give an accurate estimation of Rosaria biomass. However, the measurements of carbon contents of Rosaria are limited to only a few taxa and samples. So this is why first, before moving to UVP work, the first objective of this talk is to use chemical measurements on living specimens. And with that, we investigated the relationship between carbon content and size for a broad diversity of Rosaria. So these results have been published in limnology and oceanography. And the second part of my presentation will be about how we used the UVP5 to estimate the global contribution of Rosaria to carbon biomass and flux attenuation. So to answer to the first question, we collected Rosaria specimens in 2021 in the California current ecosystem. So we collected them in various environmental conditions by using nectars. And then we isolated and pulled together several specimens belonging to the same taxa. And for each individual, we took pictures so that we could get size measurements from these pictures. And then each package of individuals was filtered, and we analyzed the filters using the CN analyzer to get their carbon content. So in total, we collected more than 2,000 specimens belonging to various radiolarian and fluidarian taxa. So you have an overview of all the taxa that we sampled here. So these specimens range in size from 200 microns to more than several millimeters. And in total, these 2,000 specimens were pulled in 153 samples for which we analyzed the carbon content. And to these samples, we added 103 samples from literature data. So here, we then investigated the relationship between the equivalent spherical diameter that you have here on the x-axis against the carbon content on the y-axis. And we tested the relationship between size and carbon contents. And so we found that carbon content could be well predicted from cell size. Then what we did is to compare the exponents of this allometric relationship with, to the exponents of the allometric relationship established previously for all the other protists. So from the paper from Mendenhauer and Lessard. And we found that the exponent for Rosaria was significantly lower than the exponent for other protists. So what does this mean is that for the same increase in cellular volume, the carbon content will increase less for Rosaria than for other protists. So the previous relationship would give an overestimation of the carbon content of large Rosaria. So now what we will do is to use this allometry to predict the carbon biomass from size measurements obtained with the UVP. So here you have various result pictures, and they're equivalent in UVP5 images. So there are about 30 morphotypes of large resortia that we can identify on these images. So they can be assigned, for example, to the order or to the family level. And what you have to remember is that for one UVP image, you have one resortia specimen, one size measurement, and one depth measurement. So here we used a global UVP5 dataset, which was collected from 64 cruises from 2008 to 2021. And this dataset, compared to the previous abundance I showed you, includes more profiles in areas which were previously undersampled, such as, for example, high latitude areas, in particular the Southern Ocean, and some upwelling areas. So there is a total of more than 167,000 resortia specimens, all manually validated. And most of the many of the profiles allow to have images down to the mesopelagic layer. So we divided the dataset into two layers, the epipelagic layer and the mesopelagic layer. And we actually have more resortia images in the mesopelagic layer, even though we have less profiles. So then we use the size measurements from the UVP to convert them to carbon content using the relationship I showed you previously. And then to investigate their role not only in stocks, but also in biogeochemical processes for Feodaria, only because we know that they're strictly heterotrophic and that they rely on sinking particles. We use the equation from Stucker et al. To estimate their carbon demand, so their carbon requirements from their carbon content and from turnover time values from the literature. And then for each variable, we average individual biomass and demand for each layer. For each 1 degree longitude times 1 degree latitude cell of the global grid. So then each biomass value for a point of the grid was associated to a set of environmental data from the World Ocean Atlas, such as, for example, temperature or salinity. And we could build boosted regression trees to model the relationship between the observed response and environmental variables. So after that, we use again the boosted regression trees with all environmental variables to predict the response variable for all the points of the grid. So these maps show the predicted carbon concentration, so in microgram of carbon per cubic meter, in the epipelagic layer and in the mesopelagic layer for all resortia together. So what we can see from this model is that the carbon biomass is distributed worldwide in both layers. And in particular, if you pay attention to the color scale, what you can see is that this carbon concentration is about the same order of magnitude in the mesopelagic than in the epipelagic layer. And so also we, compared to the previous estimates, we also revised downward the biomass estimates. So now we found that it is 1.7% of mesozooplankton biomass in the top 500 meters. Yes, sorry. Okay. And also we looked at the integrated biomass according to latitude. And what we can see is that the biomass is actually also important at high latitudes. So for both the epipelagic and the mesopelagic layers. So these results contrast with the previous estimates I showed you, which showed a decline in biomass at high latitudes. So our models also enable to observe two different patterns and major discrepancies in the global distribution of heterotrophic and mixotrophic taxa. So mixotrophic radiolaria are mainly found well, they're found basically everywhere except at high latitude, but in particular, they are quite abundant in oligotrophic gyres. And on the contrary, so heterotrophic Phaeodaria, they are mostly distributed in productive areas, in particular in upwelling zones. Now, because Phaeodaria are heterotrophic, we estimated their carbon requirements for different regions, as you can see on this figure. And we found that their highest demand was in the subarctic Pacific. But this only represents 10% of total zooplankton demand, likely because crustaceans are abundant there. And in contrast, they can represent up to 18% of zooplankton carbon demand in the North Pacific subtropical gyre. Globally, we estimated the global carbon demand to be 0.46 petagram of carbon per year. And if we compare this demand value to annual carbon export estimates, we found that this represents from 4 to 9% of this annual carbon export value, which range from current estimates from 5 to 12 petagram of carbon per year. So then we estimated this potential percentage of carbon flux attenuation for the same oceanic regions. Again, by comparing it to carbon export values that we found in the literature. And we observed important regional variability. In particular, the potential role in flux attenuation, we found that it was the highest in the Southern Ocean, which is a key area in the global marine carbon cycle. So lastly, I'll be quick on this, but it's worth mentioning that the biogenic silica production of this mesopelagic Phaeodaria, so because this production is much higher in the mesopelagic than in the epipelagic, and this is a layer where no other silicifiers are present. So even though the mesopelagic Phaeodaria production is likely way inferior to surface diatom production, what they do here is that they can recycle biogenic silica throughout the water column. And as I said, they are likely the only contributors to the silicon cycle. Throughout the ocean, throughout the deep ocean. So to conclude, the key message that I wanted to share is that large reservoirs are less carbon dense than other protists, but we showed the importance of large heterotrophic Phaeodaria in the mesopelagic layer, not only in terms of abundance, but also because they can have a substantial role in carbon flux attenuation. And as a quick perspective, so with the UVP, we only sampled the end of the reservoir size spectrums, but we could also include smaller reservoirs for which we could use additional devices, such as, for example, the FlowCam or the ZooScan. So these small reservoirs were not included in this analysis, but they may be more abundant and maybe have higher metabolic rates, higher turnover times. So they could contribute differently to, in particular, to carbon recycling. But this remains to be investigated. And lastly, I showed that we got reservoir biomass, their role in flux attenuation. And now a question is how much of the carbon which is consumed by large Phaeodaria is actually respired back to CO2, and how much would be transferred through mesopelagic food webs. But we still don't know much about their predators. And lastly, to fully understand the role of reservoir aggregates in carbon fluxes, we need to know how much carbon they transport to depth, upon depth of these organisms. And also we need to investigate the transport mediated by their mini pellets that they can produce and which can be highly abundant. So this would be a future step to estimate the role in vertical fluxes. So lastly, I would like to thank all the people involved in this work, all the labs involved in this work, and in particular the UVP5 data providers and all the people and the crews involved in data collection. And you for your attention.
Excellent. Thank you very much, Manon. Any questions from Manon? You can either type them into the chat or just raise a hand and we can open the microphone. So I've got a quick question what people are thinking. So this is fascinating. So the field areas, it's not a group of organisms I'd come across before at all. So do their distributions correlate in any way with regions of higher surface silicate in terms of, you know, whether there's a sort of a simple link between them and the environmental biogeochemistry?
Well, yes. So there has been a study before which showed that there can be, I mean, their distribution can be influenced by a biogenic silica distribution, but this is not the only parameter. Because they live in the mesopelagic, where silica is likely to be undersaturated. So, well, I mean, there's likely enough silica for them in the mesopelagic layer.
Okay, so you don't see situations like you do in the surface with diatoms, where, you know, the diatoms tend to be found more frequently in regions where, you know, the surface waters are higher in silicate.
Yeah, because, well, rhizoians were outcompeted by diatoms in the past.
Right.
For silica. So now, but yeah, in the mesopelagic layer, they don't have any competition for silica at all, because they are the only user of it.
Yeah, interesting. Very interesting. So we've still got a minute or so. Anybody got any questions that I'd like to ask Manon? And say, I'm sure, as is often the case, the question might occur to you later on. So do feel free to contact Manon after the talk. I'm sure she wouldn't mind answering any questions that you might have. If we got anything, let me just quickly check in the chat just in case I've missed anything. I can't see anything there. And so since we're pretty much at past, then if it's okay, thank you very much, Manon. Excellent talk.
Thank you.